Hi guys, welcome to the structure of the cell surface membrane. So in terms of the specification, we will be looking at the structure of the membrane, including cell surface membranes and the membranes around the cell organelles of eukaryotic cells. So we also will be looking at the key terms like phospholipids, proteins, glycoproteins, glycolipids, and the fluid-mosaic model of them bilayer. Also, we will be looking at the cholesterol. So this is the structure of the cell membrane, and all membranes around and within all cells have the same basic structure, and they are known as plasma membrane. So what's the job of those? Mainly it's to provide this boundary between the cell cytoplasm and the environment, and they allow different conditions to be established inside and outside the cell, and they also control the movement of substances in and out of the cell. So you can pause the video for a second now, draw your structure of the cell membrane, and let's have a look what are the important things here. So this is what we've got, our phospholipids. Those are the proteins which are in the membrane. So we've got the proteins embedded in the membrane. We've got the channel proteins which will go through the outer and through the inner and outer layer of the phospholipid bilayer. We've got then the proteins that will be lying on the surface of the membrane, on the bottom, on the top here. We've got the cholesterol which will add the strength. And then finally, we've got glycoprotein and glycolipid. So now, how can you recognize them really easily? So glycoprotein, okay, they are attached, okay, to the protein. So you're looking at the protein, and then glycolipid, okay, they are attached to the phospholipids. So the branches away from the phospholipids, those from the actual protein. So what's the job of those? So glycoprotein could act as a recognition site for chemicals, for example, hormones. So section 6, it's a really good example, explains many ideas about this. And glycolipids, they act as a recognition sites. Okay, so that's the structure. And fluid-mosaic model, obviously, it's easy to kind of explain this, describe, but when it comes to then past paper questions, you're losing marks because you need to then be specific what refers to fluid and what refers to the mosaic. So fluid, because the molecules are moving. So for example, you could say phospholipids are moving. So that means fluid, and the mosaic, because they are both proteins and phospholipids within the cell surface membrane. So what's the job of the phospholipids? They make the bilayer, so they allow only lipid-soluble molecules to pass through, but the water-soluble molecules won't be able to do so. And they obviously making the membrane flexible, what we've mentioned. So here we've got a question, and we need to recognize the structure, which is represented by hydrophobic part of the phospholipid. So it's B, hydrophobic, so kind of scared of water, but don't put that on your exam, are the fatty acids. Okay, so fatty acids of the phospholipid, and when phospholipids come together, they form a bilayer, and explain why for 2 marks. So here it's the arrangement that we can see. So phospholipids have the hydrophobic tail and hydrophilic head, which refers to the glycerol and the phosphate group, and hydrophobic heads are attracted to the water molecules in the cytoplasm, and the hydrophobic tails are repelled. So bilayer is formed in that manner. So here another past paper question. So there was the experiment, they applied the stain, and you need to suggest an explanation for the appearance of the cell surface membrane as 2 dark lines. So 2 dark lines, so we're looking again back on the structure. So phospholipids making bilayer, which looks like this. and those obviously lines here will be referring to the heads, okay, to the glycerol and the phosphates. So the stain binds to those. Okay, so membrane has a phospholipid bilayer, glycerol stains to glycerol to the phosphates, phosphate group on both, remember, inside and outside. So hence you've got those two lines. So finally, proteins, we've got many different proteins within the membrane, and what we've mentioned, phospholipids let through the lipid-soluble molecules, but water-soluble molecules won't be able to pass through the phospholipids bilayer, hence we need proteins. So some act as carrier proteins to transport the water-soluble membranes, and the other ones will act as channel proteins. So channel proteins are the one here that we will use for the active transport of the sodium and potassium ions, for example, across the membrane. So here we've got another question. So we've got to complete the table, okay, which will be the channel protein and which contains only the elements of carbon and hydrogen. So elements of carbon and hydrogens will be letter D. Why D? Because those are fatty acids, and you should remember from section 1, fatty acids are made of the long chains of carbons and hydrogens, and channel protein, it's a B, because channel protein will be the one that will allow the substances to be transported from outside to the inside or all the way around, so has to go through the whole bilayer. Cholesterol finally, so the cholesterol, it's a molecule which reduces the fluidity and gives the strength to the cell membrane. And we've got another question to describe the model illustrated in figure 1 and explain the effect of higher percentage of cholesterol. So model, okay, proteins are within the phospholipids bilayer, so a mosaic and fluid because they are moving, what we've got in our notes already, and explain the effect of cholesterol. So again, keywords, so make the structure less fluid and more rigid, so provides more support strength. Glycolipids and glycoproteins, we already looked at those, so they are the ones that are attached, glycolipids attached to the phospholipids, to the glycerol, to the phosphates, and glycoproteins attached to the protein, so they provide structural support. Functions, there are many functions of the cell membrane, but I would like to show you the past paper question for your model answers, because we could ask you about the function of proteins, so enzymes, antigens, receptor carriers, channels, but here are the features of the plasma membrane. There are plenty, and I would recommend you know those in the link. So phospholipids, so, okay, linked by so, forms a bilayer. Bilayer is fluid, so can bend, okay, to take different shapes. Channel proteins, so let water-soluble molecules to pass. Carrier proteins allow facilitated diffusion. Active transport, remember, carrier proteins only for active transport, not channel proteins. Surface proteins, okay, so cell recognition, okay, and cholesterol, so regulates fluidity, increases stability of the cell membrane. So here we've got many substances can enter and leave a cell, and describe how this is possible. For 5 marks, so this is introduction for our next topic, for the all types of the transports that you need to know. What's really important, okay, you need to use the words down or against the concentration gradient. Don't just say from higher to lower concentration, because you will not get a mark. Okay, so diffusion down the concentration gradient, small non-polar molecules, lipid soluble, can pass through the phospholipid bilayer. Large water soluble has to go through the proteins. Water by osmosis, active transport, forget that, against the concentration gradient, okay, active transport only through the carrier proteins and requires ATP. Right, so that's everything for the cell membrane. See you later.